Okay, let's see. Thank you. Thanks, Yasuo. And thanks for the invitation to talk today. Mike did a really good job earlier of setting up, I think, a lot of the motivation for our lab. We're really a synthetic biology group. We're looking to make different molecules using microorganisms and other things. This talk really is about how we've adapted CRISPR-Cas9 tools to engineer Yarrowia lipolytica. I first just wanted to say a little bit about maybe why, why we're here, or why I'm here. We were lucky enough to get a DNA synthesis award from Yasuo's group, and they've been working on building a whole genome-wide, or a genome-wide CRISPR tool for us, right? And so we've provided them a list of all these targets, or sgRNA sequences, that cover the whole genome and 6-fold coverage, so 6 targets per gene. And they've made this library for us already, and we're starting to test it now. I hope to come back next year and share a lot more with you about this and how it's progressing. This is just a little bit of preliminary data that shows that the library is actually working, right? So this is really a whole disruption, or a genome-wide disruption library, and so I hope to share a lot more with that next year. Let's step back a little bit and say a little bit about the organism that we're working on. This is Yarrowia lipolytica. So this is an oleaginous yeast. It's very good at converting different sugars into lipids, right? And so that has a lot of nice biotechnology applications here. There's been some demonstrations in industry. Let's find this. Industry that have made omega-3 fatty acids here, different carotenoids and lycopene, a lot of tags as well. You can make about 90% of dry cell weight as tags. And so really we can convert glucose or some other sugars into lipids. But what this means though is also there's a really high acetyl-CoA pool. And so what we'd like to do is then adapt this organism to take this pool and convert it into any compound that we want. I'll show you a little bit of data how we produce lycopene, but other groups across the country and across the world are also making different products as well. So this is our organism of choice, and really what we need to do here, what the challenges are, is that there's a really limited set of standardized synthetic biology tools, right? How do we manipulate the genome, or how do we make edits to the genome? How do we knock down genes, or knock down transcription, or activate cryptic genes, right? And I'll show you how we've adapted CRISPR-Cas9 technologies to do all of these, right? This is also really the base technology that we've then passed to Yasuo's group, or at least this CRISPR system, to then target every gene in the genome. Okay, so this is probably a redundant slide. I imagine most people know this now. I'll include it here for completeness, is that really what is CRISPR-Cas9, right? So Cas9 is an endonuclease that will make a double-stranded break in the genome. It needs then an sgRNA, or a single guide RNA, or a guide RNA of appropriate sequence to do the targeting, right? So there's 20 base pairs that will target to the site of interest, and then together this complex then makes a double-stranded break, right? You can repair that break in 2 ways. If you have a homologous recombination or a template here, they can do homologous recombination and repair the break, or if your organism, as is the case in many yeasts and certainly in Yarrowia lipolytica, if you can have non-homologous end joining, repair that break as well, right? And so in this case then, most of our work, well, we're going to try to adapt CRISPR to do this part as well, but the whole genome-wide library will work on this technology. Can we make a SNP in the genome and then have it natively repaired by non-homologous end joining, right? So to do this, we need 2 things. You need Cas9 and you need the sgRNA, right? This guide RNA, or a single guide RNA, that's really a fusion of the CRISPR RNA and the guide RNA into a single piece that will target Cas9 into the genome. So really this becomes, from an engineering perspective, a 2-component optimization. You need both of these things in an organism of your choice if you're adapting CRISPR to it. You know, which to tackle first, right? And so we thought, or at least we, you know, in our review of the literature, that we really thought that the sgRNA was going to be the difficult challenge for us, right? Cas9 we thought we could express. We did a codon optimization of it for Yarrowia, and so we were pretty confident that we could express Cas9. The difficulty then, or at least our review of the literature a few years ago, suggested that this sgRNA expression was going to be challenging, right? And so we really tackled this problem by, or I didn't, but a student who worked on this, Corey Schwartz, and he's done a lot of the work that I'll talk about today. He designed a series of different promoters, right? RNA polymerase II promoter with 2 ribozymes on either side, and then a whole series of RNA polymerase III promoters. And so these RNA polymerase III promoters one is SNR52, which is a typical one that was demonstrated in Saccharomyces first, as well as a tRNA from glycine, which is also a polymerase III promoter. And then concatenations of this tRNA with different native RNA polymerase III promoters. And so this is just the fusion of those promoters, making that kind of a hybrid synthetic promoter. And really what we were trying to do here is scan these different promoters or other constructs as well to see if we could really make a high level expression of sgRNAs, right? Thinking that's the critical piece for us to move forward with this system. Again, we thought we could express Cas9 pretty readily, and sure enough, that was true. We were expressing Cas9 reasonably well, but what the bottleneck here was the sgRNA expression. And so here we're just showing data at day 2 and day 4 of outgrowth, so after you do the transformation of this plasmid-based system, how much efficiency of disruption of a single gene could we do? We targeted PEX10. This is a beta oxidation gene that would allow us to screen a phenotype where we could then see growth on glucose, but then not growth on oleic acid, right? So by knocking out PEX10, we would see no growth on oleic acid, and so then we could readily screen a number of colonies to determine the efficiency here. Okay, and so what we saw then is that one of these promoters, SCR1 concatenated with tRNA from glycine, gave us a pretty high efficiency of cutting, right? And so we could then, this really was our first demonstration that we could have it effectively, Cas9 effectively cut inside the genome. So what the problem here is, and I think this is not probably talked about enough, is that it doesn't always work, right? We need a number of, you usually need to screen a number of different sgRNAs or target sites before you can find an effective cutter. In the case of the last slide, we just picked one sgRNA and we tried that one and optimized for one, right? But and really in this case, this is showing some lipolytica data as well as a similar set of data that we've adapted the same system for K. marxianus. This is a high temperature yeast that we're also working with. And really what this shows is the cutting efficiency of different sgRNAs for the same site, right? So here there's tryptophan and then histidine and some others. And so this is 1 to about 4 or 5 sgRNAs per gene, right? Some of them cut well and some of them don't cut at all. Right? And so this is really, I think, the challenge moving forward is how do we really start to identify what is a good sgRNA and what is not. There's a lot of scoring algorithms for these, and we've used these to guide our kind of design of the SGs, but we don't really see a correlation yet between cutting efficiency in Yarrowia or in K. marxianus and those algorithms that are out there. The Broad has a couple and some others as well. So there isn't a real clear correlation for design here, and so this will be a bit of a problem for us going forward, and I hope that the genome-wide work that we're doing with the JGI will help us identify some of these as well. Okay, so it doesn't always work. The first tool that we wanted to develop was how can we readily put a gene into the genome, a heterologous expression cassette, and if we wanted to do some pathway design like Mike talked about earlier, how do we rapidly put a gene into the genome? And so knowing that not every sgRNA is going to cut well or not every site in the genome is really good to insert, we scanned 20 different sites hoping for good integration or a strong integration into a small subset of those that we could then use as standardized sites. What are these sites that we can repeatedly go into at high efficiency and then characterize those sites, make a nice set of plasmids that will allow us to do a quick integration of a different pathway or iterate through a pathway. So we picked these 20 different sites. Some of them are pseudogenes within the genome. Others were ones that we would regularly knock out when we were developing a pathway. And so across all of these, we picked the 5 that worked reasonably well and characterized them further. So in each of these 5 genes, this is AXP, XPR2, A08, D17, those 2 are pseudogenes, and MFE1 is a beta oxidation gene that we would readily knock out. We can get into all of these at about 50% efficiency with a GFP expression cassette. Integration of GFP into those sites doesn't really affect or doesn't affect growth at all. So that's a benefit for us, or at least one of the criteria that we want to use to identify these sites. We were surprised to find here that expression of GFP at each of these sites didn't vary, right? Or at least in 4 of them. In the last, in MFE1, we saw a little bit lower expression there, but the others all had consistent expression, so that was nice. So this gives us a set of 5 loci that we can go into readily to do pathway development. So to demonstrate this tool or to show that we can actually do something reasonably useful with it, we went about as quickly as we could really to design a different pathway or design an optimized pathway for lycopene production. And so these 5 sites allowed us to then put in 4 different heterologous genes or an extra copy of a couple homologous genes and then some others necessary to make lycopene. So we would then produce lycopene or do a design cycle using these standardized sites to integrate genes reasonably quickly, and we could show then that adding 2 and then 3 and then 4 and then 5, et cetera, the optimization from the wild type to this optimized strain, we got about 10-fold more lycopene, or at least from the start. Right, and so these sites then will allow us, or it has now allowed us to do some other pathway development a little more rapidly than we could, right? These are integrations right into the genome, so we're not plasmid-based expression anymore. We can now iterate through a pathway design in the genome, right? And this is a nice picture of a bioreactor making the lycopene in the end. This is a nice red color. Lycopene is typically isolated from tomato, and so we can now see this kind of tomato color in the bioreactor. Okay, the next tool, or the second or the third tools that we're really developing here is CRISPR interference, or CRISPRi, right? This is a targeted knockdown strategy. How do you target to the promoter of a gene, whether it's a heterologous promoter that you've integrated or a native one? How do you target to that gene and knock down in reasonable efficiency? And so what we wanted to do here, at least in the study, was to try to do a blind screen, right? To say, okay, can we target to 2 sites in the promoter and then have that without further optimization, know that it's really going to knock down the transcription of that gene. And so what we did is target one, based on some literature studies, we targeted the transcriptional start site, or dCas9 to the transcriptional start site, and then as close as we could also to the TATA box of that promoter. So dCas9 is just a couple of mutations into Cas9 that then renders its endonuclease function inactive, right? So this is really just a steric block. Can you put down dCas9 onto the promoter and prevent transcription or reduce transcription? We really then wanted to do this, kind of without knowing a priori which sgRNAs would work. Can we target 2 important sites, or 2 sites that we thought were important? Again, the TSS and the TATA box. And so what we wanted to do in terms of this test was say, can we do homologous recombination better, right? Into the genome, so we targeted non-homologous end-joining genes, and as well as some cell cycle stalling genes, or some cell cycle genes, so we could stall, or our thought was we could stall the cell in a phase that would allow homologous recombination to happen a little bit better. And so these 3 genes, Q70, Q80, and DNL4, these are genes that do non-homologous end joining, and then this series here is also suspected that we can stall a cell cycle. This blind screen allowed us to knock down 8 of these 9 with reasonable efficiency, and as the data is shown here, we got at least 50% knockdown of each of these genes, and in only one case did it not really work very well, and so this is pretty promising, right? Our blind screen worked, which is you could take 2 sgRNAs, regardless of their efficiency, and target them to the transcriptional start site and to the TATA box, and get, at least on your first pass, get pretty good suppression or repression of that gene, right? We saw then if we suppress these that each, or in a couple of cases, just in Q70 and Q80, we had an increase in homologous recombination, and this in this case was a markered one where we had a Ura3 cassette there as well. And so really then we picked these 2 and we said, okay, well, we've done this blind screen. We've targeted one to the TATA box and one to the TSS. Are both of these doing all the work, or is just one of them doing the work? And that was the kind of the second question we asked here, do we really need them both, right? And so I think the answer here is that it kind of depends on the system, or it depends on the gene and the promoter. In one case, only one, or in the KU70 or the Q70 gene, it was only one that was driving the effect. In the case of Q80, both were kind of driving the effect. Right? And so this would be found to be a little bit different across all of the set here, is that in some cases it was one driving and the other we needed both of them there. So in this case, we wanted to optimize the system overall and demonstrate also some multiplexing. So we took the sgRNA that was really doing all the work for Q70 and both for the Q80, put those together into a multiplex system, and it also added MXI1, which is a repressor, and fuse that to dCas9, and so we see then we can actually get quite substantial repression here, and that homologous recombination goes up quite a bit compared to the control. right? And these values that we're getting for homologous recombination here with 1,000 base pairs of homology regions on either side of the gene, these are approaching that what we can get or what we see with knocking out Q70 itself, right? So this repression overall is, I think, quite strong. Okay, the last tool that I want to show or to demonstrate that we've now generated here is that can we activate genes using the same type of system. We scanned a series of different activation domains, VP16, 64, and the synthetic one, VPR. This was, I think, demonstrated a few years ago by the Church group. If we've used that to DCAS9 and then you target to the promoter region, can you activate a gene, right? And so in this case, we did a similar kind of scanning across the promoter to see which position is best to activate. Here we use the GAL system from Saccharomyces, which isn't present in Yarrowia, so this is a nice orthogonal system, really. So we scan across from pretty far up in the upstream activating sequence all the way down to the core promoter, and we can see that as you get closer to the core promoter, you see some activation. Maybe here, number 5 is just a bad SG. Not sure, but at least there's some positional dependency here in activation, right? So that's nice, and at least in the non-targeting, we also see some no activation. So this is really a synthetic system, but what we wanted to do, use this system to demonstrate or to see if we could activate cryptic genes or genes that are not typically upregulated during growth. And we wanted to see if we can then push growth on cellabiose, right? So there's 2 genes within the lipolytical genome that will, there's 2 beta-glucosidase genes that will allow you to grow on cellabiose, but they're not typically activated. You can grow on cellabiose and it won't grow, and repeatedly kind of culture in there, and you can't get them to activate. And so really what we wanted to do was target these genes or the promoter regions of those genes to see if we could naturally activate the genes and then provide some growth on cellabiose. And sure enough, the answer is we can. We scanned across that promoter region as well, the native promoter region, and we found here that in both cases only one of the SGs was really doing any of the work, or that we could only really get activation in a similar position. So this is position 5 here. It's very close to the promoter, the core promoter region, just upstream of the core promoter region in the upstream activating sequence. And so in both of those cases then, if we multiplex those, we combine those into a multiplex system here and targeted them both at once, we see dramatic upregulation of those genes, about 100-fold over the native for BLG1, both in the single and multiplex case, and a little less than that for BLG2, which is the intracellular one. And sure enough, we can now grow on cellabiose. And so the non-targeting, we get almost no growth on cellabiose, and over a few days, we can get pretty strong growth on cellabiose as the sole carbon source. With that, I think that's the end. I do want to make sure, a little quick summary, that really what we're driving at here is this is the core technologies that we want to now build this genome-wide system, or the JGI is helping us to do. That the system now is going to allow us to, or at least the core promoters that the promoters that we've been showing, will allow us to now look at a genome-wide system and start to screen for different phenotypes, et cetera. So I think what we've done here is just started to develop some standardized genome editing tools. We can do CRISPR-I, CRISPR-A, and hopefully next year I can share a lot more genome-wide data. I do want to make sure that I thank Corey Schwartz. He's really generated most of this data, and he's been a senior student in my lab doing this work. With that, I'll take any questions.